Hi, welcome to Education Wave Success Made Easy Sexual Reproduction in Flowering Plants. The topic we need to cover in this video is microsporangia. Now, microsporangia and pollen grains. In the last video, I told you about how this whole bilobed structure of anthers, like this. I could draw here. This bilobed structure carries four thecas, right? These are the four thecas, and we also call it microsporangia because it act as a sac. It act like a bag so that the pollen grains could be stored in it, right? So this microsporangia, when the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangia. What it is trying to tell you is that this anther, if it is young, at the very young stage, the four thecas we have it here, the space is filled with some tissues, or you could say that the homogeneous cells, they look very similar. You could see here, all these cells are sporogenous tissues. Okay, so this is sporogenous tissue. Now, as the anther develops, so as the maturity goes on, these sporogenous tissue, this particular cell, this sporogenous, all these cells will undergo meiosis. They all will undergo meiosis, and they will meiotically divide into four micro. So it is very simple how we have this anther which is bilobed, and also in that you have four thecas, and the one theca which you could zoom it here, like you could see the structure here, that it has these little cells inside it, and these cells are going to meiotically divide. The meiosis will take place, and you will get four microspore tetras like this, and all these four microspore tetrads will separately, these all four cells will form pollen grains. So all these particular cells are capable of forming four pollens. That's why the anther is filled with so many pollen grains, right? So how these pollen grains are coming, how this much amount of pollen grain is coming from these sporogenous tissues. So the microsporogenesis is a process where the cells of sporogenous tissues, these little cells will undergo meiotic division to form microspore tetrads like this, and they all will form pollen grains, right? So this is the sporogenesis. Now, each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. So, each cell, this whole thing is sporogenous tissue, right? And each cell, this particular cell, this cell is capable of forming four pollen grains. So, you could see how this much amount of pollen grains are able to collect in this little anther, right? So each one is a potential pollen. Now this question comes in exam, that what is potential pollen? We call this sporogenous tissue potential pollen because it has the potential to develop into a pollen grain, or we could also call it microspore mother cell. Okay, so you could see here, this complete thing is sporogenous tissue made up of little cells, and these little cells are capable of forming, this one cell is showing here. Okay, this is only one cell, not the whole tissue. This one cell is capable of going undergo meiosis, and this meiosis, for example, how meiosis occurs? One cell undergoes reductional division, two cells. Then again, meiosis number two will happen, and these two cells will undergo again reductional division, and this is how this 4 haploid microspores develop. And this will undergo changes, like the single cell, this single microspore will undergo maturity again, and this is how we are going to get this structure: a generative cell, a vegetative nucleus. This is a structure of pollen grain. We'll discuss it later. Now this is the complete structure, how this whole anther from the very early stage to the maturity, this whole thing is filled with little pollen grains, right? Now see, it is genuinely surrounded by four walls layers. So if I could tell you that this structure, this porogenous tissue is surrounded by four layers. So one layer is epidermis. Okay, so this layer is epidermis. Now the second layer is the endothecium. So this is the second layer, which is known as endothecium. Now the third layer, middle layer. This is the middle layer. So this is the whole middle layer thing. Now the last one and the very important one is tapetum. I have already told you why tapetum, this thing is tapetum. So this tapetum, in NCRT, this question has asked that why tapetum cells are having two nucleuses inside it. So I have already discussed it in the last video, although I have to discuss it in here, but yeah, you could see it in the last video. The outer three wall layers perform the function of protection. So the three layers are contributing for the protection purpose only, right? But you could see that, as I told you, this tapetum has a special function here for providing nourishment to the developing cells. See how these porogenous tissue, this whole tissue thing, how each and every cell will undergo meiosis, and how these meiosis, like meiosis 1 and 2, they will form four microspore tetrads, and these four microspore tetrads will further develop into pollen grains, functional pollen grains, right? So for this functional pollen grain to develop, this whole thing needs energy. And how are they getting this energy? This tapetum possess this. Okay. So tapetum possess dense cytoplasm. This is very important and generally have more than one nucleus. It nourishes the developing pollen grains. Okay. Now you could see here this is epidermal cells and this is endothecium. Now these are the middle layers. And here we have the fourth layer, tapetum, and these are the, okay, so we have the tapetum here. Okay, okay. So this is mother cell, microspore mother cell, or we could call it potential pollen. And this will undergo meiosis 1 and meiosis 2 to form the microspore tetrad, right? So the structure of microsporangia, the process of formation of from a pollen mother cell through meiosis is called microsporogenesis. So this single microspore mother cell having ploidy, it is a diploid cell. This is also important, knowing the ploidy of a cell. How the meiosis divides the cell into two the equal cell. Okay. Now the meiosis 2 also divides them into four microspore tetrad, but the ploidy also reduces. Now, each microspore has haploid number of chromosomes. So, starting from the diploid number of chromosome and ending has the haploid number of chromosome. Now, you could see here the haploid, the microspore tetrads are going to get mitosis, like only the nucleus will divide and it will, I will discuss it later, but you could see that it is going to be forming generative nucleus or a pollen tube nucleus. Okay, so as the anther matures and it dehydrates, the microspore dissociate from each other. So this structure is going to be like this. All will dissociate. How they are going to dissociate? On maturity, they will dehydrate. and they will develop into pollen grains. Inside each microsporangium, several thousands of microspores of pollen grains are formed that are released with the dehiscence of anthers. So I told you that the anthers, this bilobe structure had a groove on it, and that groove was like a cut so that we could release the pollen grains from it. Okay. So on maturity, the all pollen grains are going to release from the anther. So thank you so much for watching. Subscribe, comment, and like. Thank you so much.